Hello students, welcome back. Today we are going to discuss one more concept from sexual reproduction in angiosperms. The concept is post-fertilization events. After double fertilization, that means the two sets of fertilization, syngamy and triple fusion, together called double fertilization. After double fertilization, the following events occurs in a flower. They are development of endosperm, development of zygote, formation of seed, formation of fruit. So today's part we can discuss about development of endosperm and development of zygote. Development of endosperm. Endosperm develops from the triploid primary endosperm nucleus by mitotic divisions. The development of endosperm begins just before the embryo development. Why it begins before the embryo development? Because endosperm provide nourishment to the embryo. That is reason why the endosperm develops earlier than embryo development. The development of endosperm could be of following three types. They are nuclear endosperm, cellular endosperm, halobial endosperm. Nuclear endosperm. In nuclear type of endosperm development, the primary endosperm nucleus divides by repeated mitotic free nuclear divisions without the formation of walls or without cytokinesis. It results in the formation of a large number of free nuclei in the center of the embryo sac. A big central vacuole develops in the embryo sac, pushing all the nuclei to the peripheral cytoplasm. Let's see the photograph. So it is a embryo sac, and inside this is the PEN, primary endosperm nucleus, and this black colored spot is the embryo. And this nuclei undergo free nuclear divisions. That means free nuclear division nothing but the nuclear division does not follow the cytokinesis. So the nuclei, first mitotic division gives the two nuclei, and it repeats, nuclei will increase, and a large number of free nuclei formed at the center of the embryo sac. Now all these nuclei are pushes towards the peripheral cytoplasm due to development of a vacuole. Now finally the cell wall formation takes place from the periphery of the embryo sac towards the center, leading to formation of cellular endosperm tissue. So this type of nuclear endosperm we can found in maize, wheat, rice, coconut, etc. So here it is a special. The coconut water from tender young coconut is an example of free nuclear endosperm. The surrounding kernel, white part, is the cellular endosperm. We can observe in coconut both free nuclear endosperm and cellular endosperm. See this photograph here. So now, if you are imagine it is a coconut and the kernel, that means the periphery, this white portion is the cellular. So nuclei separates with the cell wall formation, cellular endosperm, and the central free nuclear division. This is embryo, and these are the free nuclei. It is in coconut water. Tender coconut water consists free nuclei without cell wall formation. Later, it becomes cellular endosperm after cytokinesis. Another type, cellular endosperm. The primary endosperm nucleus divides and every nuclear division is followed by cytokinesis, making the endosperm cellular from the beginning. So, the major difference between the cellular endosperm and nuclear endosperm is nuclear endosperm is formed by free nuclear division and it becomes cellular at the end of the divisions. Whereas in cellular endosperm development, every nuclear division followed the cytokinesis or cell wall formation. As a result, from starting onwards, the endosperm is cellular. So remember that at the end, all the endosperms are cellular, but depending on the divisions, that is whether it continue along with the cytokinesis or without continuing with cytokinesis, based on that, nuclear endosperm, cellular endosperm, in this both the types, at the end endosperm becomes cellular. This type of cellular endosperm we can found in Datura petunia. Let observe the photograph. So embryo sac, pen, primary endosperm nucleus. So nuclei is divides and it forms two nuclei. Immediately it is separated by cell wall. All. So then it is considered as cellular endosperm. Another one, halobial endosperm. The endosperm is intermediate between cellular and nuclear types. So this type of endosperm development is intermediate between the cellular and nuclear types. Let's see. The division of primary endosperm nucleus is followed by wall formation. The first division followed by wall formation, and as a result, two chambers, micropylar chamber which is present towards micropyle and chalajal chamber which is present towards chalaja are formed. The chalajal cell does not divide again and it functions as haustorium. It is nothing but a slender projection, developed as a slender projection, and helps in the absorption and transport nutrients to the endosperm. Nucleus of micropylar cell in micropylar chamber divides by repeated free nuclear divisions, and further development takes place in the same way as the nuclear endosperm. Let's see the diagram. See, primary endosperm nucleus, it undergo mitotic division. Two nuclei are formed. It follows cytokinesis. So nuclear division following cytokinesis is cellular type. So the first mitotic division is followed cytokinesis. As a result, two type of cells are formed, that is micropylar cell and chalazial cell. So micropylar cell is nothing but it is present towards the micropyle, and chalazial cell which is present towards the chalaza. And this chalazial cell does not undergo any division. Later it is developed into a slender projection, and it helpful for absorption of nutrients to supply the endosperm. And this micropylar cell, nucleus of this micropylar cell undergo free nuclear divisions like first type, free nuclear divisions. So number of nuclei formed and vacuole is developed, then they pushes to the peripheral, and this peripheral nuclei are undergo cytokinesis, and it finally become cellular endosperm tissue. So the remaining micropylar cell division is same as the nuclear endosperm. Example, it is more prevalent in the monocotyledons. Some terms related to endosperm. Ruminant endosperm. Mature endosperm with irregularity and unevenness in its surface is called ruminate endosperm. Rumination is caused by, that means unevenness or irregularity is caused by the activity of seed coat or by the endosperm itself. It is found in about 32 families of angiosperms. And examples, that is Anonaceae, Palmae families. Mosaic endosperm. In some cases, the tissue of endosperm is not homogeneous, uniform, but there are patches of different colors. Such type of endosperm is called as mosaic endosperm. This mosaic word generally we can use mosaic floor at our houses. It was observed by Weber in maize plant. In maize endosperm, red and white patches appear irregularly distributed. That's why the endosperm is mosaic endosperm. In petunia and tomato, endosperm shows two types of tissues. Some tissues consisting of diploid cells and some triploid cells. These two type of cells intermix to form mosaic. So examples already mentioned in the statements itself: maize, petunia, tomato or the example, and this mosaic endosperm is first observed by Weber. Xenia. The effect of pollen on endosperm is called as xenia. This term was given by Fouquet. Example, maize. Metaxenia. The effect of pollen of somatic tissue lying outside the endosperm is known as metaxenia. This term given by Swingle, and it will observed in date palm. Development of zygote. The zygote develops at the micropylar end of the embryo sac. Most of the zygotes divide only after a certain amount of endosperm is formed. It is an adaptation to make sure that the developing embryo has enough nutrition. Already we discussed this point. Endosperm development begins earlier than development of embryo because it is an adaptation to make sure that the developing embryo has enough nutrition. That nutrition is provided by endosperm. The zygote forms an embryo by mitotic divisions. The early stages of the embryo development, called embryogeny, are similar in both monocotyledons and dicotyledons. In both, the early development of embryo is same. That early developments of embryo is called as the embryogeny. So simply we can say embryogeny is similar in monocots as well as in dicots. What is embryogeny? Early stages of embryo development. The sequence of events are given below in the development of zygote. The zygote divides transversely to form two cells. The cell towards the micropyle is the suspensor cell and the one furthest away is the embryo cell. The embryo cell divides mitotically and gives rise to structure called proembryo. The proembryo undergo a number of divisions in various planes. It goes through various stages like globular, heart shape, etc. and then develop into a mature embryo. All these sequences we can observe in the following diagram. So the different stages in the development of dicotyledonous embryo. Let's see that. So the embryo, it is generally formed in the micropylar region. It undergo first mitotic division to form two cells. The cell which is present towards the micropyle region is suspensor cell, and the cell which is present towards the chalaja, it is the embryo cell. So now here the word proembryony, that means embryo, early embryonic stages. Up to here the developments are similar in dicots as well as in monocots. So now during these developments it is undergo globular embryo and then it is become heart shaped embryo. The remaining chain of cells formed by division of suspensor cell is called the suspensor with a basal cell below. So here it is a suspensor cells. It is the basal cell, suspensor cell. The uppermost cell of suspensor. This is the suspensor. Suspensor cell undergo mitotic division to form a chain of cells. The chain of cells are called as the suspensor. Now the uppermost cell of suspensor, the uppermost first cell, uppermost cell, it is called as the vesicle cell, and lowest cell called as the hypophysis. So lowest, so this is the basal cell region and this is the lowest cell. That lowest cell is called as hypophysis. This hypophysis form as a radicle and a root cap. Radicle is the root part of the embryo, develops from this hypophysis. As the suspensor elongates, it pushes the embryonic cells deeper into the endosperm to get nutrition. A few cells of embryo nearest to suspensor develops into hypocotyle and radicle, that is hypophysis region. Other cells gives rise to epicotyle, plumule, that is a shoot region, and cotyledons. A mature embryo may have one or two cotyledons. If it is monocot, it consists only one cotyledon. If it is dicot, two cotyledons are present. The plumule is present at the apical end and the radicle is towards the micropylar end. As the embryo develops, the integuments, ovule is surrounded by covering called as integument cells, become lignified and form a covering called testa or the seed coat. So this is the developmental stages in the monocotyledonous embryo. So up to few divisions, that is embryogenic, early embryonic divisions are similar in the monocots and dicots, and later it will changes in different forms. Structure of dicot embryo. Dicot embryo consists of an embryonal axis and two cotyledons. Let's see the diagram. So these are the two cotyledons. These one, two cotyledons and embryonal axis. Portion of embryonal axis above level of cotyledon is called epicotyle. So this region is called as the epicotyle region. Epicotyle has an apex called plumule. The apex region of epicotyle is called as the plumule. It will develop as a shoot system. The portion of embryonal axis below the level of cotyledons are called as hypocotyle region. So embryonic axis above it is called as apex region is called as epicotyle and it has a plumule, and below that cotyledons, below the level of cotyledons, that is called as the hypocotyle, and this hypocotyle has radicle and root tip radicle, and this radicle is also known as root tip, and this root tip is covered by a root cap. Structure of monocot embryo. Monocot embryo consists of an embryonal axis and one cotyledon. So this is the diagram. So it is a only one and only cotyledon. That cotyledon in monocots are called as the scutellum. The single cotyledon is known as scutellum. Scutellum is present on one side of the embryonal axis. It is present only one side. The portion of embryonal axis above the attachment of scutellum, are called as the epicotyle, and that epicotyle consists the shoot apex called as plumule, and this plumule is surrounded by a protective sheet of a foliar leaf-like structure called as coleoptile. This is the plumule region. It is protected with a leaf-like foliar structures called as the coleoptile. It protects the plumule. Shoot apex is nothing but plumule. The portion of embryonal axis below the attachment of scutellum is called as hypocotyle region. This hypocotyle has radicle as well as root cap. And this root cap is enclosed in an undifferentiated sheath called as coleorhiza. And now in this entire diagram there is a one more part that is called as the epiblast. This epiblast is nothing but reduced second cotyledon in monocots. Epiblast, this we can see in the grasses. Thank you. We can discuss one more concept in the next part, that is formation of seeds as well as formation of fruit.